Welcome back, everyone, to another deep dive. This time, we're going to be talking about epileptogenic brain lesions.
Oh, wow.
Yeah, so these are basically the parts of the brain that can trigger seizures and ultimately lead to epilepsy.
Right.
We're going to be talking about this study that analyzed over 1,300 brain tissue samples.
Yeah.
It's the largest study of its kind. And the researchers are really trying to figure out, what causes these lesions on a genetic level.
I mean, it's incredible, the scale of this research. It's really groundbreaking
Absolutely.
for the field.
What exactly did they look at?
So this deep dive really focuses on something called lesional focal epilepsy, or LFE for short.
Okay.
And it's often linked to, these malformations that happen during brain development.
Yeah. Could you expand on that a little bit? What do you mean by like malformations?
Sure. So imagine during brain development, the brain is wiring itself up, and sometimes that wiring uh, doesn't quite form correctly.
Hmm.
And that can lead to these malformations, these structural glitches.
Yeah.
That can cause problems later on.
So what kind of malformations are we talking about specifically?
Well, the study focuses on two main types. MCDs, which stands for malformations of cortical development,
Okay.
and LEATs, which are low-grade epilepsy-associated tumors.
Those sound pretty intense.
They can be.
Hmm.
But think of MCDs, like I said, structural glitches in the brain's wiring,
Yeah.
while LEATs are more like benign growths that can disrupt brain activity.
Okay, that makes sense. I'm starting to get the picture here. But why is this particular study such a big deal? There's got to be something special about it.
Oh, there definitely is. See, even with surgery, which is sometimes used to treat LFE,
Right.
a lot of cases still remain a mystery.
Really?
Yeah, we don't always have a clear genetic explanation for why these lesions form in the first place.
Hmm.
So this study was trying to fill in those gaps, to really understand the root causes of LFE at the genetic level.
So it was kind of like a genetic detective hunt.
Exactly.
And did they find anything?
Oh, they did, and their findings are pretty amazing.
Awesome. I'm ready to hear about this.
Well, first of all, they were able to confirm 12 genes that we already knew were linked to LFE.
Okay, so that confirms what we already knew.
Yeah, but here's where it gets really exciting. They also discovered two completely new genes that are associated with LFE.
Wow. Two brand new genes.
These genes are called DYRK1A and EGFR, and they could potentially be used as diagnostic biomarkers, to help identify LFE earlier,
Oh, that would be amazing.
or even maybe as targets for new therapies.
Wow. So like we could potentially develop treatments that specifically target these genes.
Exactly. That's the hope.
That's huge.
Yeah.
Let's break that down a bit. Can you tell me more about DYRK1A first?
Absolutely. So DYRK1A, this one seems to be particularly linked to MCDs.
Okay.
Those malformations in the brain's wiring we talked about. And it plays a really crucial role in brain development, especially for these cells called oligodendrocytes.
Oligodendrocytes. I'm not familiar with those.
Right.
What do they do?
So oligodendrocytes are basically the support system for our neurons.
Oh.
The cells that transmit signals in our brain. They make sure that neurons can send those signals effectively.
So they're kind of like the maintenance crew of the brain.
Yeah, you could think of it that way.
So if DYRK1A is malfunctioning, it could mess up that whole support system.
That's exactly right. It's like, if the maintenance crew goes on strike, the whole system starts to break down.
Right. And that could lead to these MCDs.
Exactly. Now, there's another interesting thing about DRK1A.
What's that?
The study found that it's connected to a pathway called the MTOR pathway.
MTR pathway.
Yeah, think of the MTOR pathway like a cell's growth control system.
Okay.
If DYRK1A isn't working properly, that system can go haywire, leading to uncontrolled growth in the brain.
So basically the cells start dividing and growing too much.
Yeah, and that can contribute to the formation of these MCDs.
That makes sense.
Yeah.
Okay, so that's DYRK1A. Now what about EGFR?
Oh, EGFR is a fascinating one. You might have heard of it before, actually.
Yeah, I think it's involved in some cancers, right?
You're absolutely right. It's often found in these really aggressive brain tumors like glioblastoma.
Oh, wow. So this gene is a troublemaker.
It can be, but what's surprising here is that the study found EGFR mutations in lower grade tumors.
You mean the LATs?
Yes,
Yeah.
exactly, the LEATs. This suggests that it might play a different role in LFE compared to, its role in those more aggressive cancers.
So it's like it has two different personalities depending on the context.
In a way.
Yeah,
That's so weird.
it is. And there's another interesting observation about EGFR.
Okay, what is it?
The study found that the LEATs that were associated with EGFR mutations, they had these unusual growth patterns.
What do you mean by unusual?
Well, it's like they were spreading out differently than usual, extending into spaces where they don't normally reach. So the EGFR mutations are not only changing the genetic code, but they're actually changing the physical structure of the brain.
That's right. And this suggests that EGFR might be playing a big role in how these lesions develop and behave,
Hmm.
potentially impacting how severe the LFE is and even how well treatments might work.
This is getting really interesting. We've got these two new genes, potentially new ways to diagnose LFE and maybe even new avenues for treatment.
Mm.
But I have a feeling this is just the tip of the iceberg, right?
Oh, you're absolutely right. There's a lot more to this story. These genes and the pathways they're involved in, they're all interconnected like a complex web. And that's what we're going to unravel next.
Okay, I can't wait to dive deeper into this. It's some serious brain-bending stuff.
It is, but we'll make sure to break it down in a way that's easy to understand.
All right, I'm counting on you.
You got it.
So we've got this complex web of genes and pathways that are all involved in LFE.
Yeah.
How do these new genes, DYRK1A and EGFR, fit into all of this?
Well, that's what's so fascinating about this study. What they found is that both of these genes actually interact with other genes that we already knew were linked to LFE.
So it's not just about these two genes in isolation. It's about how they're all connected.
Exactly. It's like, imagine a network of roads connecting different cities.
Okay,
Each gene is like a city, and the roads are the pathways they use to communicate with each other.
got it.
So if you block one of those roads, traffic gets backed up and things don't run smoothly.
Right.
It's the same with genes. When a gene malfunctions, it can disrupt the entire network, which can then affect brain development and potentially lead to LFE.
So it's like a domino effect.
In a way, yeah. But it makes it really challenging to study and understand because you have to consider all of these different factors.
I can imagine. It's like trying to solve a giant jigsaw puzzle where the pieces are constantly moving.
It's a great analogy.
So how did the researchers actually study this network? I mean, did they just look at the genes?
They did a lot more than that. They actually combined genetic analysis with histopathology.
Histopathology.
Yeah, it's basically the microscopic examination of brain tissue.
Oh, wow. So they were actually looking at the brain tissue under a microscope.
Exactly. They wanted to see the physical structure of the brain and how it was affected by these genetic mutations.
That's really cool. So what did they see when they looked at the brain tissue of people with these mutations?
Well, first of all, they were able to confirm the initial diagnosis for each patient.
Meaning they had LFE.
Right. But they also observed some interesting things that were specific to the DYRK1A and EGFR mutations.
Like what?
Well, for example, every single person with a DYRK1A mutation showed signs of something called the MTOR pathway being overactive.
The MTR pathway, isn't that the cell's growth control system we were talking about?
Yes, exactly. And they could actually see that it was overactive in the abnormal cells of the brain lesions.
Oh, wow. So they could see the effects of that traffic jam we were talking about.
Precisely. It was like having visual confirmation that DYRK1A was disrupting the MTOR pathway.
That's pretty amazing.
They also saw some interesting patterns with EGFR.
Okay, tell me about that.
So the LAAs that were associated with EGFR mutations,
Uh-huh.
they had these unusual growth patterns.
Unusual how...
They were extending into spaces where they don't normally reach.
So the mutations weren't just changing the genetic code, they were actually changing the physical structure of the brain.
Exactly. And that's really significant because it suggests that EGFR might be influencing how these lesions develop and behave.
I see. So it's not just about identifying the genes, it's about understanding what they're actually doing in the brain.
You got it. This study is a great example of how we can combine different research approaches to get a more complete picture of what's going on.
This is really groundbreaking stuff. So we've learned about these two new genes, their potential role in LFE, and how they can actually change the structure of the brain. But there's got to be more to the story.
Oh, absolutely. There's a whole other layer to this research that we haven't even touched on yet.
Oh, really? I'm intrigued. Tell me more. It's fascinating to think about these MCDs and 8s, and it makes you wonder if there's a connection between them. Like, could there be some kind of underlying thing that goes wrong during development that leads to these different types of brain lesions?
That's a really interesting question, and it's something that researchers are definitely looking into. This study actually provides some clues that there might be some shared mechanisms at play.
Oh, really? How so?
Well, remember that gene PIK3CA that we talked about earlier?
Yeah, I think you mentioned it's involved in a bunch of different conditions.
Exactly. It can cause a whole range of different problems depending on where and when it mutates during development.
So, like, the same gene can have totally different effects depending on the context.
Precisely. And that suggests that even though MCDs and 8s seem very different on the surface, they might actually share some common developmental roots.
Okay, I'm starting to see the connection here.
Think of it like a tree with different branches.
Okay.
Each branch represents a different brain condition, but they all come from the same trunk, right?
Right.
So the trunk represents those shared developmental pathways, and if something goes wrong in the trunk, it can affect all the branches in different ways.
That's a great analogy, and you can really visualize that. So where do we go from here? What are the next steps in LFE research?
Well, there are a couple of things that need to happen. First, we need to validate these findings in larger groups of patients.
Right, because this study was pretty small.
Exactly. It was a great starting point, but now we need to see if these results hold true in a larger population.
Makes sense.
And we also need to dig deeper into how DYRK1A and EGFR actually contribute to LFE.
So it's like we have the suspects, but we need to gather more evidence to build a case.
Exactly. We need to figure out the mechanisms, the step-by-step process of how these genes are actually causing these brain lesions.
And that will hopefully lead to new treatments.
That's the goal. And it's important to remember that it's not just about DYRK1A and EGFR. This study highlights the importance of looking at the entire network of genes and pathways involved in LFE.
It's a complex puzzle.
It really is.
Right.
But with each new piece we uncover, we get closer to understanding how this condition works and how to treat it.
Well, this has been an incredible deep dive. We've learned so much about these new genes, their potential role in LFE, and the importance of understanding the complex network of genes involved in brain development. For anyone listening who might be affected by LFE, what message would you like to leave them with?
I think the main takeaway is that there's hope. This research shows that we're making progress in understanding this complex condition, and that's really encouraging. And with every new discovery, we get closer to finding new treatments and improving the lives of people with LFE.
That's a great message to end on. Thank you so much for joining us on this deep dive. It's been a fascinating journey into the world of brain science. And to all you listeners out there, remember, every new discovery brings us closer to unraveling the mysteries of the brain. Until next time, keep those brains buzzing.